smFISH is known for measuring the absolute number and subcellular location of mRNAs. Our two-color smFISH protocol has the added capability of measuring the kinetics of transcription and mRNA degradation.
Using this method, many samples can be processed simultaneously without extra time or effort. For example, 4 time-course experiments yielding 48 samples can be processed for imaging in 8 hours. Our protocol is broadly applicable for many genes and bacterial species.
To prepare cover slips and glass slides for the experiment, use forceps to place cover slips and slides in a Kaplan jar containing 100% ethanol, and place the jar in a water bath ultrasonicator for 15 to 20 minutes. At the end of the sonication, wash the slides and cover slips 3 to 4 times with ultra-pure water before refilling the jar with 70% ethanol and repeating the sonication. When all the sonication steps are done, dry the cover slips and slides with nitrogen gas. Place the cover slips into an empty 1,000 microliter pipette tip box and place the slides into a clean slide box. Then, following the holes of the pipette tip box, use a hydrophobic marker to draw circles onto the cover slips and apply a 20 microliter drop of 0.1% poly-L-lysine to each of these wells. To set up a time-course experiment, add 750 microliters from a 20 milliliter exponentially growing E. coli culture to a 1.5 milliliter culture.